Hello everyone, my name is Golara Javadi from Robotic and Control Lab at University of British Columbia. Our paper is Toward-targeted ultrasound-guided prostate biopsy by incorporating model and label uncertainty in cancer detection. Prostate cancer is a global health challenge. 1 in 7 men in North America will be diagnosed with this cancer during their lifetime. Blood test and digital rectal exam are the first series of tests to check for prostate cancer. If prostate-specific antigen goes beyond a certain level, or digital rectal exam shows some abnormality, considering the patient age, it will be flagged and be required to undergo the biopsy procedure. The current gold standard for prostate cancer diagnosis is the histopathologic analysis of specimen taken during the biopsy procedure, guided by transrectal ultrasound. In this process, 8 to 12 samples are taken from predefined location of prostate gland, and this is clearly a blind and not patient-specific process, which can result in over or underdiagnosis of the cancer. Another method, which is more patient-specific, is to detect the suspicion region by multiparametric MRI. This technique has high sensitivity but low specificity for identifying the intermediate risk or low volume of disease. Therefore, the fusion of multiparametric MRI and ultrasound has been used to improve the prostate cancer detection. It basically enables targeting of predetermined cancer location in multiparametric MRI during the transrectal guided biopsy. This means that the biopsies is still required with a suspicious NP-MRI. In addition, registration of 3D transrectal ultrasound to NP-MRI is a complex task, and it fails to fully account for patient motion or organ deformation occurring during the biopsy. So with all the issues in the current protocols, an ongoing challenge in prostate cancer patient care is the ability to distinguish aggressive cancer that has a very high likelihood of leading to metastasis from less aggressive or indolent cancer and benign tissue. Using the current technology, we are dealing with a huge amount of unnecessary biopsies and, of course, patient discomfort. As one of the possible solutions for these challenges, we are proposing an ultrasound-based method, and our ultimate goal is to show the cancer probability map. This column map can be used for decision-making during the biopsy procedure and improve the accuracy significantly. And of course, eventually, it tries to decrease the high rate of unnecessary biopsies. To this end, we take advantage of a tissue typing approach called temporal enhanced ultrasound data. TEAS is defined as the time series of RF echo signals obtained from a fixed region over a multitude of transrectal ultrasound frames. The whole idea behind this technique is to use a machine learning approach to learn differentiating features between the benign and cancerous RF time series, and finally generating a cancer probability map for biopsy guidance. So as mentioned in systematic biopsy, targets are obtained from predefined prostate zone. During the biopsy session for RF data acquisition, we ask the radiologist to hold the ultrasound transducer still for approximately 5 seconds while we capturing 200 frames of RF data. After saving this data, the biopsy gun will be fired to collect the tissue sample. Later, we identify the location of the biopsy according to the needle tip and obtain the spectral feature of corresponding RF time series. This will actually generate our dataset with 183 cores from 41 patients in total. And we divide our data into mutual exclusive training and test set. On the other end, the biopsy samples will sent to the lab and labeled by pathologists. The pathology report comes back with percentage of cancer in core, often called as involvement, and the grade of the cancer for the entire biopsy core. But the special information on the tumor location is not specified in this report. So based on these available histopathology results, we will label our data to identify if a core is cancerous or benign. For this work, we employ a two-dimensional unit architecture proposed by Sedghi on similar datasets for assigned class probabilities to each pixel in our data. Since the ground truth histopathology information is only available for biopsy cores and not for the entire image plane, we generate labels for defined region of interest. The size of this region is same as the needle core and include 2 by 80 mm area determined by the tip and orientation of the biopsy needle. The generated label are 1 hot vectors for each pixel, which represent their class. So here we depict the deterministic prediction of network for every patient in tested. In this figure, all cores for a patient are depicted in one column. Each core is labeled with its histopathology verified glycemic score or tissue type. Blue and green represent the correctly predicted cancer and benign cores, and the red cores are the ones with the wrong prediction from our model. So now the major research question is how can we automatically identify the misclassified cases? So we know there are two main sources of error in a classification approach. The first error, referred as epistemic uncertainty, is generated by the network due to uncertainty in model parameters and overfitting to data. Therefore, this error can be estimated by varying the model. MC dropout is one of the most popular methods which applies dropout at test time and measure differences in model output to quantify the uncertainty. We choose this approach for calculating the epistemic error. The second type of error, known as aleatoric uncertainty, and it's related to inconsistent labeling and image noisiness. Since these errors are originated from a measure of genuine uncertainty in a data, aleatoric uncertainty is unlikely to be reduced with increasing training data, and it can be estimated by varying the data. So one approach to estimate this uncertainty is to vary the data at test time or test time augmentation. So this approach measures differences in output across augmented versions of the same data. And for this work, we apply the impeding augmentation method, where square patches of data are masked to generate new augmented data. And finally, we use a color map generation module to create the cancer likelihood map and high uncertainty region. So in a well-calibrated method, we expect the accuracy versus confidence plot to be monotonically increasing. And our experiment shows similar behavior for aleatoric uncertainty, as you can see in this plot, which shows the accuracy versus confidence. However, the MC dropout for estimation of epistemic uncertainty did not follow this trend. And our conclusion is that MC dropout is not a good approach with our dataset. We hypothesize that the main reason in this phenomena here is the non-stationary nature of our data. So you can also refer to the details of experiments in our paper. Here we show the cancer likelihood map overlaid on prostate B-mode ultrasound images. The top row shows the binary prediction of cancer versus benign within the prostate boundary. The red and blue colors refer to cancer and benign prediction, and the middle region is shown with a green boundary. So we show the high uncertainty regions with the purple in the bottom row, and here we have two cancer cases. The first one has low uncertainty and it's been correctly identified as cancer with the model. And the second one has high uncertainty around the needle location, and it's been falsely predicted as benign with the model that we have. So we can see how the high uncertainty maps can show us which results and output of a model are reliable. Here we estimate the model and label uncertainty in classification of prostate cancer from ultrasound data. And we use test time augmentation method to estimate the label uncertainty, and we apply MC dropout method to estimate the model uncertainty. So we demonstrate the benefit of an uncertainty color map visualization to identify the potential failure cases for model prediction. So as a future work, we will involve a clinical study to demonstrate that such visualization will impact the clinical decision making to improve the cancer detection. And for another extension to our work, we will consider other methods of epistemic uncertainty estimation, such as deep ensemble models, to achieve a better calibrated estimation of model uncertainty. This work is the result of collaborative effort consisted of technical and clinical experts. And I'd like to thank our team and funding agencies for making this work happen. Thank you everyone for listening.